Parkinson's disease may not start in the brain, but rather in the kidneys, according to new evidence uncovered by researchers in a study published in Nature Neuroscience. The team found that a key protein involved in Parkinson's, called alpha-synuclein, can build up in the kidneys and travel to the brain through nerve connections, especially when kidney function is poor. This shifts the focus away from the brain as the starting point and suggests that chronic kidney disease might silently trigger changes that later affect the brain. Alpha-synuclein is known to cause damage when it misfolds and clumps, forming toxic deposits seen in Parkinson's and similar disorders. In tissue samples from people with Parkinson's or kidney disease, the researchers found these misfolded proteins in kidney nerves and, in some cases, even in brain regions linked to the disease. In animal tests, healthy kidneys cleared the protein quickly, but in mice with kidney failure, the protein lingered and eventually spread to the brain, where it caused damage and movement problems. The researchers also showed that cutting the kidney's nerve supply blocked this spread entirely. Additional experiments confirmed that red blood cells, which carry much of the protein, may worsen the problem when kidney disease is present. Mice with blood cells that did not produce alpha-synuclein had less brain damage, unless the harmful protein was directly injected. The overall findings suggest that when kidneys cannot filter properly, alpha-synuclein can build up, deposit in nerves, and travel to the brain, setting the stage for Parkinson's. This could explain why people with chronic kidney disease face a higher risk. However, not all cases of Parkinson's begin this way, and the kidney-brain link may not apply to everyone. More studies are needed to see how this process works in people over time. Mice with two fathers have now had babies of their own, marking the first time male-only genetic material has produced fertile animals that can reproduce. In a new study from Shanghai Jiaotong University, scientists used sperm from two male mice to create embryos, removing the female DNA from eggs and editing specific DNA control regions to fix issues that normally prevent embryo development. These regions are called imprinting control regions, and they usually need a balance of male and female gene activity to work properly. Out of 259 embryos implanted into female mice, 3 mice were born and 2 survived to adulthood. Both of those later had healthy pups of their own. The success rate is still very low, but it proves that with precise gene editing, it is possible to produce fertile offspring from 2 male parents only. This process is more complex than creating offspring from 2 females, which has been done before with fewer edits. To create mice with 2 fathers, researchers use a multi-step process combining gene editing and stem cell techniques. They started by collecting sperm from 2 male mice. 1 sperm cell was used to create lab-grown stem cells, which were then edited using CRISPR to disable around 20 genes that normally need input from both a mother and a father. These edited cells were combined with a second sperm cell and inserted into an egg that had its original DNA removed. The embryo, now carrying only male genetic material, was placed into a surrogate female mouse. Out of 164 embryos implanted, 7 live pups were born, and now some of them have their own babies, showing that these mice are capable of further reproduction.
A female organ that's often considered useless may actually support ovary function and fertility, according to a recent study published in eLife. The structure, called the rete ovarii, was first discovered in humans over a century ago, but was long dismissed as a leftover from development with no real purpose. Today, biologists are re-examining the structure and its possible importance. New research in mice shows that the rete ovarii plays an active role in maintaining the ovaries by directing protein-filled fluids and responding to hormonal signals like estrogen and progesterone. Scientists found that the rete ovarii is made up of 3 distinct regions and that it produces important proteins that may help with egg cell development and hormone regulation. The structure also seems to work like an antenna, sensing changes in the body and sending signals to the ovary to support its function. This network of tubes may help create the right environment for healthy eggs and proper ovarian activity. While these findings are based on mice, researchers believe the same could be true for humans, since the rete ovarii is found in many mammals, and early reproductive development is very similar. Understanding this overlooked organ could open new paths for treating fertility problems, ovarian diseases, and better understanding how the female reproductive system really works. Scientists now hope to study whether removing or damaging the rete ovarii directly affects fertility, which would confirm just how important this long-ignored structure really is.